This video tutorial is all about microorganism cultures. Specifically, it looks at how to culture microorganisms effectively using aseptic techniques. Microorganisms will grow on almost any material that provides the carbon compounds for respiration and a source of nitrogen for protein synthesis. However, in the laboratory, microorganisms are usually grown in one or two types of growth media. The first is a set jelly-like substance called agar, which is melted and poured into Petri dishes. Typically, nutrient agar contains peptones, yeast extract, salts, and water, and might also contain sort of glucose or blood. The second is a soup-like liquid called a broth, which is kept in bottles or tubes. Aseptic techniques have been developed to reduce the likelihood of contaminating the medium with unwanted bacteria or fungi. The standard procedures include washing your hands, disinfecting the areas, having a Bunsen burner operating nearby to heat the air. This will cause the air to rise and prevent airborne microorganisms setting, and also creates an area around it of sterile air in which the microbiologist can work. As you open a vessel, if you're using that, you pass the neck of the bottle over the flame to prevent bacteria in the air entering the bottle, and the bottle should also actually be flamed as it's closed. You don't lift the lid of the Petri dish off completely. You just open it enough to allow the sort of introduction of the desired microorganism. And any glassware or metal equipment should also be passed through the flame before and after contact with the desired microorganisms. Now, microorganisms can grow almost anywhere. The key is to ensure that they grow on the medium used and that you grow the desired microorganism rather than anything else. You don't want to infect your medium by mistake. Now, growing microorganisms on agar plates involves three main steps: sterilisation, inoculation, and incubation. So first of all, sterilisation. The nutrient agar medium and any equipment to be used must be sterilised. The medium is sterilised by heating in an autoclave at 121 degrees C for 15 minutes. The high temperature is achieved by boiling water under high pressure inside the autoclave. This kills all living organisms, including any bacteria or fungal spores. When the media has cooled sufficiently to handle, it's poured into sterile Petri dishes and left to set. It's important that the lid is kept on the Petri dish to prevent infection. Inoculation is the introduction of microorganisms to the sterile medium, and this can be achieved in a number of different ways. Firstly, streaking. So you get a wire inoculating loop, and you use that to transfer a drop of liquid medium onto the surface of the agar. The drop is then drawn out into a streak by dragging the loop across the surface. You just got to make sure and you take care not to break the surface of the agar when you do that. Secondly, seeding. So a sterile pipette can be used to transfer a small drop of liquid medium to the surface of the agar or to the Petri dish before the agar is poured. Thirdly, spreading. So a sterile glass spreader may be used to spread the inoculated drops over the surface of the agar. And finally, a small cotton swab or cotton bud can be moistened with distilled water and used to collect microorganisms from the surface and then carefully wiped over the surface of the agar medium. The Petri dish, once it's been inoculated, must be labelled, and the top is then taped to the bottom using strips of adhesive tape. You do need to be careful not to seal the Petri dish completely, as this will lead to selection of anaerobic bacteria, which can be pathogenic. The Petri dish is then placed into a suitable warm environment, such as an incubator, and it's placed upside down as this prevents drops of condensation falling onto the surface of the agar. It also prevents the agar medium from drying out too quickly. Suitable temperatures will depend on the type of organism being grown. And I'm just going to sort of mention a few of those now. First of all, looking at the bacteria. So bacteria tend to grow into visible colonies, which can be shiny or dull. Some colonies are round with entire edges, whilst others can have crenated edges. The colonies can also be a range of different colours, and each colony will result from a single bacterium. In contrast, fungi, so filamentous fungi, grow into a mass of hyphae, which may also be circular, but the mass is not shiny and often looks a little bit like cotton wool, with sort of furry aerial hyphae. Single-celled fungi, so like yeast, grow as circular colonies. All Petri dishes must be completely sterilised after use and before disposal. Thoroughly washing your hands is also absolutely fundamentally important after handling a Petri dish, as any moisture coming out of the Petri dish could be a source of infection. And finally, just think about using liquid medium instead. So a liquid broth is initially clear but will turn cloudy when bacteria have grown. A liquid broth can be useful to increase the numbers of microorganisms before transferring to agar plates for counting or for identification. Similar aseptic techniques will be used as described sort of already in this tutorial. Liquid broth can be used to investigate sort of population growth.